Usually when we talk about a medical diagnosis, there's this expectation precision, right? Like it feels like engineering. You break your arm, you go to the hospital, the X-ray shows that jagged white line on the screen, and the doctor just points at it and says, there it is. That's the problem.
Right. It's very binary. It's either broken or it's not.
Exactly. It's clean. And frankly, I mean, it's comforting. But then you step into the world of neurodevelopment and pediatric brain cancer, and suddenly that X-ray machine feels completely useless. Like we're looking at a diagnostic landscape that is just incredibly murky.
Oh, it really is the absolute definition of diagnostic muddy waters. I mean, when you're dealing with the developing brain, you are looking at billions of cells trying to wire themselves together in real time. So when something goes wrong, it's rarely just one clean break.
And that is exactly where we're going today. Welcome to this deep dive. Our mission today is to take a pretty dense, highly technical, but honestly groundbreaking molecular biology paper recently published in the journal Cell Death and Disease and extract the real aha moments for you. We're going to explore a microscopic tug-of-war happening inside cancer cells. And by the end of this, we're going to uncover how a single tiny fat molecule acts as like a master on-off switch for tumor growth.
It's a remarkably elegant study, really. To set the stage for you, we're zeroing in on a disease called medulloblastoma.
Right.
This is the most common malignant pediatric brain tumor out there. It makes up about 20% of all childhood central nervous system cancers. But the tricky thing about medulloblastoma is that it isn't just one single disease.
Yeah, the researchers focus on a very specific subgroup of this cancer today, don't they?
Exactly. We are looking specifically at what's called the Sonic Hedgehog, or SHH subgroup.
Yeah.
This specific type accounts for about 30% of all medulloblastoma cases. It originates in the cerebellum.
That's the part of the brain right at the back of the skull, right?
Yeah, exactly. The lower back of the brain. It starts there during early brain development. And, I mean, the stakes here are incredibly high. While we do have treatments like radiation and chemotherapy, they're really blunt instruments.
Yeah.
They often leave these young kids with severe, lifelong neurocognitive side effects. And to make matters worse, these specific tumors are notoriously difficult to treat because they relentlessly develop drug resistance.
Okay, so if we want to understand how to stop this tumor, I imagine we first have to understand what's actually building it, right? Like, what is the engine driving this chaotic growth? You mentioned it's called the Sonic Hedgehog pathway. And I have to admit, the name sounds like a joke.
I know, it's one of the great quirks of geneticists. The pathway was actually first discovered decades ago in fruit flies. When scientists mutated this specific gene, the fly embryos ended up covered in these little spiky denticles.
Oh, making them look like hedgehogs.
Right. So when they found the mammalian equivalent, they just named it Sonic Hedgehog. But, naming aside, this is an ancient, highly conserved evolutionary pathway. We share it with fruit flies for a reason. It is basically the master blueprint for embryonic development.
So it's essentially the instruction manual that tells a cluster of cells how to become a brain.
Precisely. It tells cells where to migrate, how fast to multiply, what type of tissue to become. But in this specific medulloblastoma, this developmental engine gets stuck in the on position. It's just blasting the multiply signal constantly.
So how does a pathway even get stuck on? Like, what does that actually look like at the cellular level?
Well, think of it like a bucket brigade of proteins passing a message from the outside of the cell down into the center. Normally, this pathway is tightly regulated at a structure on the surface of the cell called the primary cilium, which acts sort of like a tiny cellular antenna.
Okay, an antenna.
Right. When there is no Sonic Hedgehog signal around, a receptor sits on that antenna and acts like a heavy brake pedal. Everything is quiet.
But when the Sonic Hedgehog signal arrives from outside the cell, it takes the brake off.
Yes, exactly. The signal binds to the receptor, the brake is removed, and this allows a different protein further down the line called SMO, or smoothen to activate. SMO is like the green light.
Oh, okay.
It sends a cascade of signals deep into the cell that ultimately frees up our main culprit today. It's a protein called GLI1.
GLI1. Okay, so GLI1 is the final messenger, the chief operative here. What exactly is its job?
GLI1 is what we call a transcription factor. Its entire purpose in life is to physically travel into the cell's nucleus, grab onto the DNA, and turn on specific genes.
The cancer genes.
Yeah. The genes it turns on are the ones that tell the cell to divide, to survive at all costs, to multiply. Now, there are other versions of this protein in the cell that can sometimes act as repressors to turn things off. But GLI1 is exclusively an activator. It only knows how to press the gas pedal.
Okay, let me stop you there. Because if I'm listening to this, a really obvious question comes to mind. If we know that GLI-1 is the bad guy with his foot pinned on the gas, forcing this cancer to grow. Why don't we just make a pill that targets GLI-1? Like, why don't we just design a drug to block that specific protein and shut the whole thing down?
It's a brilliant question, and honestly, it's essentially the million-dollar puzzle in oncology right now. Transcription factors like GLI-1 are incredibly difficult to target directly with drugs. You can think of them as these big, flat, floppy proteins.
Speed.
Yeah, and for a typical small molecule drug to work, it needs a deep pocket or like a crevice on the protein's surface to latch into. Think of a key fitting into a lock.
Right, okay.
But GLI-1 just doesn't have good pockets, so it's often referred to in the field as undruggable.
So you can't easily jam a wrench into GLI-1 itself.
Exactly. Yeah. So pharmaceutical companies did the next best thing. They looked upstream. They thought, well, if we can't stop GLI-1, let's stop the protein that activates it.
The green light protein you mentioned earlier.
Right, SMO. So they developed drugs that target that SMO protein on the antenna, and the FDA actually approved some of these drugs. They physically bind to SMO and shut the pathway down.
Well, that makes total sense. Just cut the supply line before the signal even reaches GLI-1.
And in some cancers, like certain skin cancers, it works brilliantly. But in this pediatric brain cancer, the tumors are incredibly adaptable. When you introduce that upstream drug, the tumor mutates. The SMO protein literally changes its physical shape so the drug key no longer fits in the lock.
Right.
Or worse, the tumor finds a biological bypass route, completely skipping the SMO protein and just manufacturing massive amounts of GLI-1 anyway.
So the upstream drugs fail, the supply line is bypassed, and the cancer just keeps aggressively growing. I mean, if the factory's main power switch is broken and our drugs don't work, aren't we out of options? What else is there to even target?
That feeling of hitting a wall is exactly where the researchers of this new paper started. They realized that if you can't stop the cell from producing GLI-1, you have to look at what happens to GLI-1 after it's already been manufactured.
Okay, so this is where things take a turn. We're moving past the initial manufacturing phase and looking at how the cell handles this rogue protein once it's out in the wild.
Exactly, which brings us to a biological mechanism called S-palmitoylation.
S-palmitoylation? That is a mouthful. Break that down for me. What is the cell actually doing here?
It's what we call a post-translational modification. So after a protein is fully built, the cell can still tweak its function by attaching other molecules to it. In this specific case, an enzyme comes along and attaches a lipid, a tiny 16-carbon fatty acid called palmitate, directly onto the GLI-1 protein.
Hang on, I'm trying to visualize this. Why would the cell attach a fat molecule to a protein? Does it change its weight? Or, I don't know, does it shield it?
It fundamentally changes how the protein behaves. It anchors it, and more importantly for our story, it shields it.
Oh, it's like a video game mechanic.
You mean?
Like, imagine GLI-1 is an enemy soldier in this microscopic battle. This S-palmitoylation process is like the soldier finding a buff, like a heavy suit of fatty armor. Once this fat molecule is attached, GLI-1 puts on the armor and becomes virtually invincible inside the cell. It doesn't break down, it just marches into the nucleus and keeps yelling at the cancer to grow.
That is a fantastic way to picture it. The fatty armor stabilizes GLI-1, and the researchers wanted to know, well, who exactly is putting this armor on the enemy soldiers?
Right.
They didn't just guess either. They went into a massive international database and analyzed the genetic sequencing of over 700 human medulloblastoma samples.
Over 700. That is a staggering amount of data to sift through. What were they looking for?
They systematically looked at all 23 known enzymes in the human body that are capable of attaching this specific fatty armor. They compared the SHH brain tumors to other types of brain tumors. And when they ran the numbers, one specific enzyme stood out like a sore thumb. It was being massively overproduced in these specific cancer cells.
The armor?
Yes. An enzyme called ZDHHH13.
Okay, so this ZDHHH13 enzyme is acting as the dedicated armorer, just constantly slapping these protective fat molecules onto GLI-1.
Precisely. And they didn't just rely on genetic data. They proved it visually. They used specialized microscopes to light up these specific proteins inside living cancer cells. And they saw the armor and GLI-1 physically locked together in an embrace near the center of the cell. They proved that this armor is directly interacting with the rogue protein.
But wait, if the cell is attaching this armor, biology is usually a two-way street, right? Like, if there's an enzyme putting the armor on, there must be a natural mechanism to take it off.
You've hit on a core principle of biology: equilibrium. Yes, there are rival enzymes whose entire job is to remove that lipid armor. The researchers screened a bunch of candidates and found a specific enzyme called ABHD17A.
The armor stripper.
Exactly. This enzyme removes the fat molecule, effectively stripping GLI-1 of its invincibility, and the researchers tested this balance in the lab. When they artificially pumped more of the armor stripper into the cancer cells, GLI-1 levels plummeted.
And if they suppressed it?
When they suppressed the armor stripper, GLI-1 levels skyrocketed.
Okay, let me just make sure I have this tug of war pictured correctly for you, the listener. On one side, we have the armor enzyme, which is heavily overproduced by the cancer, constantly outfitting GLI-1 with protective fat. On the other side, we have the armor stripper trying to take it off. If I'm a cancer cell, my survival strategy is obvious. I want to flood the zone with the armor and completely shut down the stripper.
And that is exactly what the tumor does to maintain its aggressive growth. But the researchers needed to dig even deeper. They understood the who, the armor, and the stripper, but they needed to understand the how. How exactly does a single tiny fat molecule make this massive protein invincible?
Right, because he's just saying its armor is a metaphor. What is physically happening at the atomic level?
To figure that out, they mapped the entire GLI-1 protein. They chopped it up into tiny segments and painstakingly mutated different amino acids, building blocks of the protein one by one.
Wow, that sounds tedious.
It is, but they were hunting for the exact molecular coordinate where the armorer attaches the fat molecule. And they found it at a specific spot called cysteine 1034.
Cysteine 1034, okay. Why is that one specific spot so critical to the protein survival?
Well, to understand that, we have to talk about how cells take out the trash. We have a beautifully elegant system called the ubiquitin proteasome system. Normally, proteins don't live forever. When a cell realizes a protein is old or malfunctioning, or, say, telling the cell to become a cancer, it dispatches an enzyme to tag that bad protein with the tiny molecule called ubiquitin.
Ubiquitin, I've actually heard of this. It's essentially a biological trash tag, right?
Exactly.
It's like slapping a bright neon kick me sign on GLI-1's back so the cell's internal garbage disposals. The proteasomes know to grab it, grind it up, and recycle it.
That's a great analogy. The proteasome is a molecular shredder, and it only shreds things wearing the ubiquitin trash tag. Now, here is where it all connects. The region right around that cysteine 1034 coordinate is what biologists call an intrinsically disordered region. It's a floppy, flexible, highly exposed part of the protein.
Okay.
It is the absolute perfect place for the cell to attach that ubiquitin trash tag.
Wait, if that's the perfect spot for the trash tag, and that is also the exact spot where the armorer attaches the giant fat molecule.
You see it?
The fat molecule physically covers up the spot. It's like trying to slap a sticky note on someone's back, but they're wearing a massive, bulky backpack. The trash tag literally cannot reach the target.
Yes. In chemistry, we call this steric hindrance. The sheer physical bulk of that lipid armor blocks the cellular machinery from attaching the trash tag. And because the tag never gets attached, the garbage disposal ignores GLI-1. It never gets shredded. It just builds up, travels to the nucleus, and relentlessly drives the cancer forward.
That is just terrifyingly brilliant. The tumor is exploiting a physical loophole to hide its main weapon from the cell's own regulatory system. But, how did they prove this wasn't just a clever theory? How do you prove that covering up that one spot is the whole ballgame?
They proved it in two really compelling ways. First, they used a chemical inhibitor, a compound that globally blocks all fat-attaching enzymes in the cell.
Okay.
When they treated the cancer cells with this chemical, the armor couldn't be attached at all, and the results were immediate. GLI-1 levels absolutely crashed. The lifespan of the protein inside the cell dropped from over 10 hours to just 4.5 hours.
Because the kick-me signs were finally sticking and the garbage disposals kicked into overdrive.
Yes, exactly. But they wanted to be even more precise. So they went in and genetically mutated just that single coordinate on GLI-1. They changed cysteine 1034 into a different amino acid called serine.
Okay, what does that do?
Well, chemically, serine is incapable of receiving that specific fat molecule. It just won't attach to the result. That newly mutated GLI-1 degraded incredibly rapidly, and the cancer signaling was completely crippled.
Okay, so they proved the physical mechanism in a petri dish. They found the loophole. But I have to push back a little here because a plastic dish full of cells isn't a brain.
True.
A dish doesn't have an immune system or blood vessels or complex tissue. So how do we know this microscopic tug of war actually dictates the fate of a living, breathing tumor?
It's the ultimate test of any biological discovery, really. You have to prove it in vivo in a living organism. To do this, the researchers used two distinct genetically modified mouse models to test both sides of this enzyme war. Let's look at the first model, where they tested the armorer.
Okay, set the scene for this first mouse model for us.
So these mice are genetically engineered to have incredibly aggressive, constantly active Sonic Hedgehog pathways. The pathway is permanently blasting at full volume from birth. Because of this genetic drive, 100% of these mice develop severe medulloblastoma very early in life.
Wow, 100%.
Yeah, it's a devastating model. They develop cranial protrusions, loss of motor control, and severe fluid buildup on the brain.
That is an incredibly tough baseline to try and fix. What do the researchers do to them?
They went in and genetically deleted the gene that creates the armorer. They completely removed that specific enzyme from the developing brains of these mice.
So they essentially blew up the factory that makes the fatty armor, leaving GLI-1 completely exposed. What happened to the mice?
The results were striking. Now, because the upstream genetic drive in these mice was so overwhelmingly strong, they did still develop some tumors. But the severity was dramatically reduced. MRI scans showed that the fluid buildup in their brains was significantly relieved. The tumors physically shrank.
But how do you even measure what's happening inside the shrinking tumor? Do you just, weigh it?
No, they look at it at the cellular level. They take slices of the tumor and use specialized chemical stains that literally light up under a microscope based on what the cell is doing.
Oh, that's clever.
Yeah, one stain only sticks to cancer cells that are actively dividing and multiplying. When they looked at the mice without the armorer, the number of multiplying cells plummeted, and they used another stain that only lights up when a cell is in the process of dying, undergoing programmed cell death.
And I'm guessing that stain lit up like a Christmas tree.
Exactly. The cancer cells were actively dying off. And most importantly, this translated to a real-world survival benefit. By simply removing that single armorer enzyme, the overall survival of these severely afflicted mice extended significantly.
Which, in mouse time, for a cancer engineer to be that aggressive, is a massive leap.
Okay.
So that proves that taking away the armor hurts the tumor. What about the other side of the war? The second mouse model.
Right. The second model tested the armor stripper. They used a different type of genetically engineered mouse. These mice are missing one copy of that PTCH1 brake pedal we talked about at the very beginning.
Okay, so the brake is a bit faulty, but not entirely broken.
Right. This model is much slower and closely mimics natural human tumor development. Only about 15 to 20% of these mice naturally develop tumors over the course of several months.
Okay, so for these mice, the researchers went in and genetically removed the armor stripper. They deleted the enzyme whose only job is to rip the fat off the protein. If we follow the logic we've been building, if you remove the only thing capable of destroying GLI-1's armor...
What do you think happens to the cells?
The armor stays on permanently, the cells can't take out the trash, GLI-1 builds up and builds up, the pathway amplifies, and you basically push the cancer into overdrive.
Exactly. It's a biological cliff, and removing the stripper pushed them right off it. The incidence of tumors in these mice absolutely skyrocketed. It jumped from that baseline of around 18% up to nearly 46%. Oh. Yeah, nearly half the mice developed tumors rapidly. This proved conclusively that a lack of the armor stripper is enough to push vulnerable cells right over the edge into full-blown cancer.
It is a perfect, terrifying biological seesaw. If you take away the armor, aggressive tumors physically shrink and cells die. If you take away the stripper, the tumor rate more than doubles. It really makes you realize the sheer importance of these microscopic equilibriums happening inside us every single second.
It does. And it highlights why deep fundamental research like this is so critical. We started this discussion with a clinical frustration, a drug that targets the top of a cellular pathway, which ultimately fails because the tumor mutates and bypasses it. By digging down to the very bottom of the pathway, to the physical atomic structure of the final protein, they discovered a brand new vulnerability.
Which brings us to the ultimate question we always want to answer here. What does this all mean for you, the listener? Why does a deep dive into lipid armor, steric hindrance, and cellular trash tags matter outside of a biology textbook?
It matters because it provides a completely novel blueprint for fighting so-called undruggable targets. For years, the dogma was that we can't drug GLI-1 directly because it doesn't have the right shape. But this paper proves we don't actually need to drug GLI-1.
Right. We don't have to attack the enemy soldier directly. If pharmaceutical companies can design a drug that simply inhibits that armor enzyme, ZDHHE13, We can effectively strip the armor off the entire GLI-1 army.
Exactly. We don't have to invent a drug that kills the cancer cell. We just take away its shield, expose the target, and let the patient's own natural cellular garbage disposals clear it out.
It's like biological judo. You aren't using brute force, you are just using the cancer cell's own internal mechanisms against it. By blocking the armor, you bypass all of those upstream mutations that cause the traditional drug resistance. It's a completely different angle of attack.
That is a perfect analogy, and it opens the door to potential combination therapies. I mean, imagine hitting the tumor from the top with an SMO inhibitor, and from the bottom by blocking the armor. You trap the cancer from both sides.
And beyond just medulloblastoma, it leaves us with something quite profound to think about. When we hear about devastating diseases, especially pediatric brain cancers, we tend to think of massive structural failures. But this entire life-or-death battle, whether a tumor shrinks or grows uncontrollably, doesn't come down to a massive organ failure. It comes down to the addition or subtraction of a single microscopic lipid molecule sitting on a single tiny amino acid out of millions of moving parts inside a cell.
It really is humbling, isn't it? If a modification that unimaginably small can dictate the fate of a resilient brain tumor, it forces you to wonder, what other invisible microscopic tugs of war are happening inside our cells right now? What other tiny lipids or hidden trash tags are dictating our aging, our health, or our susceptibility to disease in ways we haven't even begun to discover?
A completely murky diagnostic landscape entirely controlled by the beautiful, invisible precision of a single fat molecule. Keep that in mind the next time you think about how your body works. There is a whole universe of battles happening right now, just beneath the surface. Thanks for joining us on this deep dive.